The main role of antibodies secreted by plasma cells is to tag foreign molecules or microbes for destruction. Binding of antibodies to antigens can trigger several effector mechanisms. Neutralization occurs when antibodies bind to antigens, blocking their activity and making them harmless. For example, antibodies cover the surface proteins of this virus, preventing it from hooking up with the host cell. Other antibodies may bind to toxin molecules on a bacterium. The bound antibodies attract macrophages, which dispose of the invaders by engulfing them, a process known as phagocytosis. Antibody molecules have at least two binding sites, allowing them to form a clump of microbes by binding to two antigens on two different cells. This process, called agglutination, traps microbes and makes it easy for macrophages to capture them and dispose of them by phagocytosis. Precipitation is similar to agglutination, except this process clumps together free antigen molecules dissolved in body fluids. This inactivates them and attracts macrophages, which destroy them by phagocytosis. One of the most important mechanisms of humoral immunity is the activation of complement proteins. Binding of antibodies to a foreign cell triggers complement proteins in body fluids to form a hole in the membrane of the target cell. The hole allows water and solutes to enter the cell. The foreign cell swells, bursts, and dies. Humoral immunity, mission accomplished.